Our next talk is by Professor Marinka Zitnik. So Professor Marinka Zitnik is an assistant professor at Harvard University with appointment in the Department of Biomedical Informatics, Broad Institute of MIT and Harvard and Harvard Data Science. Her research recently won Best Paper and Research Awards from International Society for Computational Biology, Bayer Early Excellence in Science Award, Amazon Faculty Research Award, Rising Star Award in Electrical Engineering and Computer Science, and Next Generation Recognition in Biomedicine. Being the only young scientist with such recognitions in both EECS and biomedicine. Surely she is one of the rising stars in this area, and her topic is infusing structure and knowledge into biomedical AI. Over to you, Marinka.
Hello, everyone. Thank you so much, Niloy, for this kind introduction. Thank you to the organizers for this wonderful conference. It's really a delight to be here with you today. So I will talk about infusing structure and knowledge into biomedical AI. I'm sharing my screen as well, so I hope you can see the first slide, and we'll go from there. So the papers, data sets, and code that I will be describing and highlighting throughout the talk are all linked from our lab website. If you go to zitniklab.hms.harvard.edu, you will find pointers to code bases, data sets, and more resources. So with that, let's begin. The first question that I'd like to ask and kind of provide my answer to, and that can stimulate the rest of this presentation, is to thought and probe a bit more thoughtfully about what makes biomedical data so unique. And I think there are a variety of aspects and characteristics of biomedical data and applications that require thoughtful and careful development of machine learning models that can then also impact ML research at the more fundamental level. So first, it's very important that for most of phenomena that we are studying in biology and medicine, it is the case that there is rarely a single data set that can capture the entire complexity of that phenomena. Because of that, there is a need to integrate heterogeneous confounded data that span from molecules all the way to the level of society. And so that is challenging because that kind of view often does not fit into a more classic machine learning data representation where we think of our input data set being a single large flat data matrix, essentially. And so that's often not the case, as we have data that are multi-scale, that describe behaviors of molecules, individuals, even entire populations or communities, data that are heterogeneous in the sense that they come from in the form of experimental readouts from biotechnological platforms, curated annotations, or even self-reported measurements. And they might be confounded in a variety of different ways as they're generated by different technologies and measurement platforms. So that's a kind of a very unique but incredibly important and central challenge in biomedical data set. The other is that when we think of machine learning on biomedical data, we often do that in order to discover some of new fundamental insights, but then also guide downstream experimentation, either in the wet lab or in clinic. Because of that, we need to have models that can translate predictions into actionable hypotheses, meaning that it's no longer enough to simply optimize models for accuracy, but there are a variety of other auxiliary criteria that are equally important, or in certain applications even more important than only optimizing for a particular, for your favorite performance metric. And the third challenge that I think makes also biomedical data quite unique together with the first two is that for problems that are very new, that are really at frontier of science, there is typically very little labeled data available. Not surprising in a way, right? So if a problem is new, of course, it's of great interest for study in order to advance that particular area of biology and medicine. But just because of that, we often have a lot of very little labeled information. And so training of standard supervised learning frameworks is then, because of that, often not feasible. And so that's a challenge because many recent breakthroughs in AI have depended on lots of labeled data being available that then allowed models to achieve broad generalization. And that is often not the case when we are tackling new problems. In particularly problems at frontier of science. And I will give concrete examples of that later in the talk. So the way we are tackling these three challenges that I just outlined in my group is through the use of networks or graphs that we see as a universal language that allow us to bring together very heterogeneous, interconnected systems and represent them as networks or graphs. And so that allows us to kind of identify and model interactions between proteins within a cell and other types of molecules within a cell, how those behaviors of molecules are changed when external agents such as therapeutics and drugs bind and target particular molecules. And so how all that interacts within a human body, within an individual that might have multiple diseases at the same time. And that individual is, of course, embedded into more broader healthcare system or entire population. And so you can see that across these different scales, there are networks everywhere because the data is so in our system and biological systems are so interconnected and intertwined. And so networks or graphs are very effective representations to bring narrow heterogeneous data together. Once we have those data sets, the question then becomes, how do we learn and reason over networks and produce very accurate predictions that we can also interpret in a meaningful way? So we have been particularly interested in developing core new graph neural network architectures over the last several years that have emerged as a leading paradigm for deep learning on networks in biology and medicine. And we have seen many areas of success across different subfields, as well as in other fields outside of biology that I believe some of other speakers in this conference will be addressing or have addressed over the last 2 days. And so very briefly, the kind of the formulation of graph neural networks that we are using and I'll be using for the rest of the talk is as follows, and here's how you can think of it. So imagine we start with an input graph given to us by a set of nodes and a set of edges. In this particular case, we have some nodes that correspond to genes and then edges would indicate physical interactions between proteins that those genes encode. And so a graph neural network can be then thought of as a non-linear deep transformation function, here denoted by F, that maps each and every node from the input graph to some low-dimensional, in particular d-dimensional vector that we refer to as embedding. So the idea here is that those embeddings should be learned such that two nodes that have similar local neighborhoods in their input graph, so on the left part of the slides, those two nodes should be represented and mapped by F to points that are nearby in the embedding space. So effectively, now this function F is optimized such that it learns an embedding space that when performing linear algebra operations in that embedding space, it reflects the structure and topology of the input graph. And so they're depending on how we define when two nodes in the input graph have similar local neighborhoods, and how that should be translated into that low dimensional space can give us a large number of different architectures. And so really in the last several years, really a number of architectures have been developed that operationalize this notion of different kinds of similarities on local neighborhoods of input graphs and different kinds of distances or metrics that define the notion of the quality of an embedding space that we're optimizing for. most predominant approach of how graph neural networks are implemented today is that we rely on the neural message passing, which is essentially a kind of network propagation on steroids that allows us to learn what is the optimal way to propagate neural messages, which are initialized as embeddings throughout the network, following edges in that network, such that we get the optimized embedding space. And that is the project that is the paradigm that powers today's graph neural networks. GNNs have really shown success across a very broad area of broad types, diverse kinds of biological networks. In particular, we and many others have demonstrated that they are very useful for learning over patient networks, where you have networks where nodes are patients connected by different kinds of relationships, such as familial relationships, medical similarities, and so on. Networks in biology, when we are concerned with modeling hierarchies of cellular systems or ontologies, disease pathways, various gene interaction networks, or cell-cell interaction networks in single-cell genomics recently, as well as knowledge graphs. Across those very diverse kinds of networks, GNNs have been quite powerful. And so with that introduction in mind, my plan for today's talk is the following. I would like to highlight two topics, and the first will be about how important it is and how it can be achieved to infuse structure into biomedical AI algorithms. And particularly, I'm interested in a very challenging setting that relates to few-shot learning for graphs. And I will describe an algorithm for that approach, and then discuss two applications that relate to drug discovery and protein biology. The second part, I will talk about infusing knowledge into biomedical AI, and particularly I will focus on subgraph neural networks as an algorithm that we recently developed, and as an application, I will talk about using SubGNN for disease diagnosis. So with that in mind, let's begin. So the first topic relates to few-shot learning on graphs. And so to motivate this project, let me spend a minute just kind of establishing the need for models that can operate and learn in challenging settings where only limited amount of label data are given. So the challenge is that existing GNNs require abundant label information in order to train accurate predictors. But what I argue is that those labeled examples are scarce in many problems that are at the frontier of science. And so examples of that include novel drugs in development, that are drugs that are not yet approved, but are actually compounds that are being investigated for their potential to become drugs. And so there is very little label data available about them. Essentially, no information about how those drugs would behave in patients, because of course they were not yet given to them. Think about emerging pathogen, novel emerging diseases that until very recently we have never seen before. So of course, there are no label data that one could readily use to train supervised learning frameworks. And many other examples exist as well, including modeling of rare diseases, identifying and diagnosing patients that are otherwise very hard to diagnose, and so on. So visually, you can think of this problem as follows. What prevailing methods assume is that our input graphs look like what is shown on the left part of the slide. We start with a graph that is already richly labeled. So here, for the problem of node classification, we see that many of the nodes on the graph, on this graph on the left part of the slide, are already labeled as indicated by their node color. So given all that information, plus the graph structure, a GNN model can train a powerful node classifier. However, what happens in the real world when we move outside of benchmark graph data sets, what we see is the reality that looks like what is shown in the right part of the slide, which is that we have these graphs that are vastly unlabeled, and there is this dark matter that represent our unknown knowledge. Most nodes have very little annotations, represented here by gray color, and only a small number of them have some labels associated with them. The question then becomes, well, how can we learn this challenging regime that is shown in the right part of the slide? So from the machine learning standpoint, that means that we need a strategy that will allow us to train and GNNs in such a way that will be able to operate in few shot learning regimes. So let me introduce or suggest a solution to this problem. So first, I will just spend one slide on background on what few-shot learning is for those on this conference who might not be familiar with this term. So few-shot learning is a particular instantiation of meta-learning, which is a broader field, and few-shot learning is an instantiation of meta-learning in the field of supervised learning. It refers to a setting where only a small number of labeled examples called shots are available for each class that we want to predict. So what I'm showing here on this slide is an example of two-shot three-way image classification. So it's not about graphs, it's for image classification, just as an example on few-shot learning. But the idea here is that it's called two-shot three-way learning because there are three classes that we're interested in predicting, and for each of those three classes, only two shots or two labeled examples are provided. So the way few-shot learning in this case would operate is through into two stages. The first stage is known as meta training phase. The second stage is known as meta test phase. In the meta training phase, we would take our model and we would train it in such a way that we would ask it to solve a large number of training tasks, where each of those training tasks is designed to be very challenging and challenge the model to learn how it and adapt very quickly to a new task with limited labeled information. So each training task here, as you can see, we have the task is to identify a predictor that can distinguish between images of three different classes, where two labeled images are given for each of those three classes. And so the hope is that we can identify a mechanism that can transfer information from one training task to the other training task, so that eventually, as the model is solving more and more training tasks, it will become better and better at quickly adapting to a new task, because it has already seen so many other tasks. And so when that stage finishes, then the second stage is the meta testing phase, which is actually the phase that is concerned with training a predictor that we really care about. So in this particular example, at test time, imagine we would want to build a classifier that would be a duck versus dolphin versus chicken classifier. And even though we might have only two images for each of those three classes, few-shot learning would make it possible to train a powerful predictor, an accurate predictor for this particular problem. So what we want to now see is with kind of this particular framing of definition of few-shot learning, well, how can we have a strategy that would allow us to train graph predictors with so few labeled data points available. And so what I will outline is an algorithm called GMeta, which is a general algorithm for few-shot learning in graphs. The details, as well as a theoretical characterization and extensive benchmarking of GMeta is described in our paper that we published at recent NeurIPS. And so the way GMeta works is that it also has two stages of training. And so in the meta training phase, we define a large number of training tasks. I'm showing here an example where those tasks are all node classification problems. But in each training task, we ask the model to learn, in this case, node classifier by that can distinguish between different between labels that are represented here by different shades of yellow. And so when the model is trained on training task 1, we define a mechanism, which I will outline next, that allows us to transfer the knowledge that it has learned from training task 1 to effectively provide initialization for training task 2. And then repeat the same process on training task 2, and then proceed to training task 3, and so on. And so that we are doing that because at meta test time, when we ask the model to build a classifier that is now need to generalize to a new label set, here represented by blue colors, that has not been seen before, the GMeta model can effectively do that with only few blue examples of blue labeled nodes. And that is because the model has been primed before on to can be effectively initialized by solving those yellow labeled sets. So a bit more formally, the way we can think of this particular instantiation of GMeta, where we would have a single graph and would be concerned with node classification task, is that imagine we have a particular graph G with some distribution of labels that are denoted by Y. So what the goal in this graph meta learning setting is that we want to identify and design a meta learner that would be able to quickly quickly adapt to an unseen label set Y star by learning from tasks from other label sets Y. And so pictorially, that means that we have a single graph, some of these examples of classes from one label set Y and others. And the goal is to use that as effectively as possible to generalize to a label set Y star that has no overlap with Y. So this is a setting of meta learning that is defined on a single graph. But GMeta is a general strategy. So for those who are interested and would look at paper, will see that it is possible to use GMeta in settings where you have a data set of many different graphs, as well as it works for any graph machine learning task, including link prediction and graph level classification. The key idea that allows GMeta to quickly adapt to a new task with very few labeled examples is the notion of local subgraphs. So the idea here is the following, that when we have our input graph with only a few labeled nodes, and we are training our neural message passing network F that will embed those nodes to d-dimensional embeddings, what we also do in addition to learning embeddings, we learn subgraph signature functions. So for each and every of the training tasks, we learn a signature that is generated by a signature function, and that signature represents and describes the task in a very effective manner. That signature function is defined by looking at a local or enclosing subgraphs around nodes that are labeled nodes in that particular task. And so every task is then represented as a form of distribution over enclosing subgraphs around nodes that are labeled in that task. And that task is summarized by subgraph signature function. So subgraph signature function is the mechanism that the GMeta uses to transfer knowledge from one training task to the other and essentially map subgraph structure to an effective initialization for a new task with a new label set. So you might be wondering, why local subgraphs? Why would that be a justified approach? So that is possible to study from the theoretical standpoint as well as empirically. So from the theoretical standpoint, we showed that the influence of one particular node in the graph on the target node that we want to make prediction for decays exponentially as we move further away from the target node. So because of that, that means that if we learn signatures of tasks, but by looking primarily around locals in the structure of local subgraphs that surround target nodes, then we will capture and preserve near the same feature information, as well as if one would look at the entire graph and try to learn signature function of the entire graph rather than subgraphs. And as a side effect, that also leads to very scalable an algorithm, because we don't need to process entire graphs, but only subgraphs. Conceptually, the way you can think of the value of subgraphs is as following. When we are learning with graphs, we can leverage two sources of predictive power. The first source of predictive power in graphs is typically label propagation. So with label propagation, the idea is that nodes that have the same label, they will be found nearby in the graph. They might be direct neighbors, or they might be relatively close to each other, reside in the same region of the graph. And so when the other source of predictive power in graphs is structural similarity, where nodes that have the same label might not reside in the same region of the graph, but they might have similar network shapes in their local neighborhoods, even though nodes might be actually quite far away from each other. So the reasoning for GMeta is then the following. When labeled information and labels are very scarce, then simply relying on label propagation will not be sufficient. When only a handful of nodes are labeled, it is challenging to reliably propagate labels through entire graphs and rely on label propagation. And so in those settings, we need to better leverage structural similarity. That is exactly what GMeta does by learning those subgraph signature functions to describe tasks effectively. So more of the details from the theoretical end about GMeta are described in our paper, but I will now move to what I think is an exciting application of GMeta to the problem in drug development, and in particular identifying the novel therapeutic opportunities for emerging diseases. So to motivate that work, I will discuss a bit the need for rapid therapeutic innovation in times where we are faced with unprecedented health emergencies. So one of them is certainly when we're faced with pandemics, they demand development of safe and effective therapies at unprecedented speed. And so that is a challenge for drug discovery and development nowadays. Why? Because the development of new drug from scratch relies on iterative cycles, experimental and clinical testing, and approval of new drugs that takes on average between 11 and 16 years and costs between $2 and $3 billion to develop a new drug from scratch. That is not feasible in times when rapid therapeutic innovation is needed. So the challenge then is how to compress, or is it possible to compress years of work into months or even weeks through the use of novel data sources and powerful machine learning algorithms. So the project that I will describe took primarily place between March 23rd last year and June 15 last year. During that span of just 3 months, we were able to identify relevant data sources, train our graph neural network predictors to identify what drugs might be promising therapeutic opportunities for a novel emerging pathogen, and then send those algorithmic predictions to experimental laboratories that validated predictions and identified drugs that were predicted that also experimentally were confirmed in human cells. And so this entire project is described in the paper that we published at PNAS earlier this year, and I will highlight it over the next few slides. So the first thing that is important to discuss is certainly what was the data set that we used for this project, and what was the experimental setup for it. So to identify therapeutic opportunities for COVID-19, we constructed a large knowledge graph, and that knowledge graph consisted of has the 4 key components. It consisted of first the human protein-protein interaction network, which is a network where nodes are proteins connected by edges representing physical contacts between proteins in cells. And so that genome-wide protein interaction network was supplemented by additional edges that linked proteins to drugs based on what are proteins that each and every drug that is approved in the US targets, and by binding to them. In addition to then drug target edges, we also had edges between proteins and diseases that indicated information about what proteins are associated with the onset of certain diseases, in the sense of when proteins are malfunction for a variety of reasons in terms of their errors in genetic code or folding, there is increasing chance that the disease will develop. And for those diseases that were common diseases with some available treatments already that existed and were on the market, we had information about, well, what drugs approved for use in the US treat what kind of diseases. So given that knowledge graph that consisted of protein nodes, drug nodes, disease nodes, and edges between them, and common disease and treatments for common diseases, the question was, can we use that to identify candidate treatments for COVID-19 in a zero-shot manner? Why zero-shot manner? Because for COVID-19, there were zero available therapeutic opportunities or drugs at that time. And so there were essentially no labeled examples available for COVID-19 class in our case, which is then a zero-shot learning or extreme case of zero-shot learning setting. So when I was talking about the view of the data set on the previous slide, with this you can be visually represented through this cartoon, where what we can see is the underlying human protein interaction graph, which is shown by nodes and edges here in the central part of the slide, and then in that human interactome or human protein interaction network, we have a large number now of information about how drugs that are approved in the US target and affect human body by binding to certain proteins, and those proteins then propagate the effects of that binding event propagate by following edges of the network, and that's how the drug exerts its therapeutic effect. And for COVID-19, the way we represented it is by relying on the information generated by Gordon et al. From UCSF, and their team has expressed 26 out of 29 SARS-CoV-2 proteins, and then used mass spectrometry to identify 332 proteins to which viral proteins bind. And so that was the information that we used to define a viral disease module or COVID-19 disease module that was a subgraph of the PPI network induced on those set of 332 human proteins that viral proteins bind to. So given that information, the way GMeta was instantiated for this problem of drug repurposing was in the following manner. During the meta training phase, we defined a large number of training tasks with the goal of the model identifying what are the underlying biological and network connectivity patterns that can distinguish between drugs that are likely to be safe and effective for a disease and drugs that will not work for the disease. And to extract those patterns, GMeta was tasked to first effectively predict treatments for diseases that we already understand well and for whom some treatments are available. So each of those training tasks was defined around a different disease. So training task 1 would be concerned with the model distinguishing between drugs that are approved for disease 1 versus drugs that are not approved for disease 1. And then training task 2 is defined in a similar way, but it's now focused on a different disease. And so we defined a large number of those tasks across many common diseases with some available treatments, so that at the meta testing phase, GMeta model was able to prioritize drugs for COVID-19, even though there were no examples of successful treatments. When we generated prediction lists, first, we wanted to understand and get some insight into, well, how trustworthy those predictions might be. And so to that end, we evaluated performance of predictions made by the algorithm against drugs that were considered in clinical trials at that time. So the results of that experiment are shown on the left part of the slide, where we see that the GMeta approach and graph neural network approaches used as individual predictors outperformed a variety of alternative algorithms based on network diffusion and classic drug repurposing predictors. So that has motivated us to really take the power of GNN predictors and combine them with more classical predictors to get a final prioritized list of drugs that could be used by our collaborators who work on the experimental side. So to that end, we teamed up with National Emerging Infectious Disease Laboratories in the US, and our collaborators there took predictions that the models made and that the model was most confident about and whose explanations were reasonable. And those explanations were identified by some post hoc explainability methods that we have developed. And so they took those top predictions and they screened them for efficacy against SARS-CoV-2, first in African monkey cells and then in human cells. The result of that process was that by screening top-ranked, top-predicted drugs in human cells, we obtained a near 62% success rate, which is in sharp contrast with 0.8% success rate or hit rate that one would get by doing the screening in a non-guided manner, in a manner that would not be assisted by the outputs of these algorithms. And so this is an order of magnitude better hit rate among top ranked drugs than what would be possible by alternative approaches. And we kind of found it particularly exciting to look at. And so then next, I would like to in the remaining time that I have, change the gear a bit and talk about infusing knowledge into the kind of the challenges and opportunities that lie when we want to infuse more of the domain knowledge into biomedical AI, in addition to inducing structure. And so to that end, I will spend a bit of time motivating the need for moving beyond classic learning tasks in graphs and what kind of new applications and problems we can tackle with those new frameworks. So the motivation for this part is the question of disease diagnosis. So the way diseases are diagnosed in clinic is typically that a human expert, in this case clinician, would make an informed decision based on a large number of different kinds of signals or information that they have available about patients. So one of the central part of that are phenotypes or symptoms that a patient has. So phenotypes are defined as an observable characteristics that typically result from interactions between genotypes, molecular behaviors within an individual, as well as the environment where that individual lives. And so physicians utilize standardized vocabularies of phenotypes or symptoms to describe human diseases. And so the question of diagnosis can then be defined as a problem of matching between now sets of phenotypes that a patient might have and a particular vocabulary of diseases that current medicine knows. And so by modeling diseases then as collections or sets of phenotypes rather than individual concepts, one can diagnose patients based on sets of their presenting symptoms, as well as other types of information that describes the environment in which a patient lives and their molecular and genetic makeup. So from the problem definition standpoint, we can formalize this idea in the following manner. So think of a graph, consider a graph G that would be built by looking at the standard vocabulary of phenotypes, where nodes in that graph would represent phenotypes and edges would indicate semantic similarity between those phenotypes. And so in that setting, a patient is given by a set of phenotypes. So a patient is a subgraph of G. And the learning task then becomes, given that graph and a large number of patients a large number of subgraphs on top of that graph, some of those patients already being diagnosed, meaning that their subgraph labels are available. The question is to predict the disease or identify candidate disease that the patient might have, defined as predicting the label that is most consistent with the phenotype subgraph. And so that effectively means that what we need to do, we need to have a model that will make predictions at subgraph level, rather than the level of individual nodes or edges. And so to that end, we have recently developed a model known as subgraph neural networks. And the details of the model are described in this NeurIPS paper, but I will just try, in the interest of time, define the problem and then leave the details, defer the details, refer interested interested in the audience to look at our paper. But the definition here is the following. We want to learn subgraph embeddings such that the likelihood of preserving subgraph topology will be maximized in the embedding space that we want to optimize for. So the idea is that we start with our input graph that consists of an underlying base graph overlaid by a large number of subgraphs. Here's an example with just five subgraphs S1 up to S5. Given that as input, we want to design a function that will embed those subgraphs such that for S sub I and S sub j that have similar subgraph topology, they will be embedded close together in the embedding space. And so what we have developed is a subgraph neural network that can achieve exactly that. It can disentangle distinct aspects of subgraph topology that we characterize in the paper and model as separate channels that capture position, neighborhood, and structure of subgraphs. And then it can embed on the fly any subgraph you would have and generate a powerful embedding that can then be fed into a downstream classifier to predict labels for those subgraphs and therefore aid in diagnosis. So perhaps I will just show one final slide of results for this particular test that I used to motivate this part, where we looked at the two problems. The first one was related to identifying neurological diseases, and the second was related to identifying a class of metabolic diseases. And what we found is that subgraph neural network SubGNN outperforms baselines by an average of 77% when looking at its performance on synthetic data sets, and by up to 125% when we look at them on real-world data sets, such as HPO Neuro and HPO Metab data sets. And in particular, the different channels that SubGNN has allows SubGNN to comprehensively consider and disentangle different aspects of subgraph topology. So we think and believe that this really creates an exciting opportunity to move away from simply performing node classification and link prediction on graph structure data sets and think of even more exciting formulations. Finally, my last slide, I promise, is the slide where I would just like to answer perhaps a question that some in the audience might have about, okay, how can one start working in the area of drug discovery and development and kind of contribute to this incredibly exciting research field that the therapeutics ML has become, when when it's hard to orient ourselves in the sense of what the data sets are available and ready for machine learning development. So to this end, we have founded an open science initiative that is called Therapeutic Data Commons. And Therapeutic Data Commons is a collection of machine learning ready data sets, leaderboards, benchmarks, data set splits, molecule generation oracles, data processors, data functions, and so on. That is an entire ecosystem that allows machine learning scientists to work on meaningful, relevant problems in drug discovery, retrieve data sets in 3 lines of codes, high quality data sets that are ready for ML scientists to be used and for method development, and then partner with domain scientists who can identify what are meaningful therapeutics tasks in order to facilitate algorithmic and scientific innovation in that space. So with that, I conclude, and I encourage everyone to look at our lab website to find more information about the projects that I highlighted, and I'll be happy to take any questions. Thank you.
Thank you, Marinka. Great talk. It's an absolutely fascinating talk and great area. So I will first ask Anirban. Anirban, you have a question.
Oh, sorry, I didn't realize. Thank you. Beautiful talk. Beautiful talk. So my question is, yeah, I think we might have lost the context because it's from a little back, somewhere in the middle of your talk. So you were designing, when you were trying to sort of do few-shot learning using graphs, one of the methods that you mentioned that you would design a bunch of sort of different problems, right, in order to do the training, right? Difficult sort of problems in order to do the training, and then do the testing, right? The few shot training, something. So how do you design these training problems? That was my question. Right? What is a nice way to go about to design the training problems so that my sort of final model is something that I really like? So
yeah, so that's a great question, and the main challenge here relates to the notion of we want to design them in such a way that we can achieve positive transfer. What that means is that essentially when we will train the model on the final task, or which is the task that we're actually interested in, we will know that performance on that task will actually be better than if we would not use few-shot learning or those pre-training tasks or these auxiliary tasks in a way, right? So and that was a non-trivial question, actually very recently, because it is hard to ensure that we always achieve positive transfer. So when that became possible was through the use of techniques and strategies that allow us to define few-shot learning problems in as two-stage problems where we are training the model in the meta-training phase. The way that is done is through using MAML, where we can essentially identify and kind of transfer information from one test to the other based on gradient optimization, rather than just manually trying to a priori identify how similar tasks are. And because of that, we don't need to manually feature engineer what the tasks are and how similar they are and how much information we should transfer from one task to the other. That is determined automatically by training the model in this meta-training, meta-testing phase through the use of MAML. And that is what GMeta does.
Thank you so much. Again, thank you for the beautiful talk.
Animesh, you have something to ask?
Yeah, so thank you very much for the very interesting talk. So I was just wondering, like, probably I missed something somewhere. So is it not the case that in general, the idea of GNNs was to learn from a very small set of examples? So even when you showed the in one of your first few slides that there are a lot of labels available for the network to learn from, but I think the idea is to only supply a few of them as training examples, and then allow the network to learn from those set of small training examples. So I was just wondering, like, how is the general GNN framework like becomes more challenging in your problem case?
Yeah, so probably what you think of a general GNN framework might be the neural message passing architecture that's actually learning how to propagate neural messages along edges of the graph. So the neural message passing on its own would not change, but you can not propagate neural messages and use neural message passing where you only have a handful of labeled nodes. Imagine having a graph with millions of nodes, and of those millions of nodes, you have labels for a handful of nodes, like 10 or 50 nodes. Propagating then neural messages along edges of the graph by relying on this small number of labeled examples is not reliable and does not only that it's not reliable, it's practically, you cannot train powerful classifier. So what the GMeta does, it says that in addition to neural message passing architecture, what we need is a way to capture more than the local neighborhoods of labeled nodes. And so what GMeta, and so to that end, it relies on structural similarity. So it is characterizing and quantifying the similarity of network shapes in the local neighborhoods of those small number of labeled nodes, so that we don't only rely on labels, but we rely on labels plus structure that surrounds the labels. And so in doing so, we have more information that we can kind of leverage and extract, that the neural message passing algorithm can rely on when training the predictor. And that is what that part of relying on structural similarity that is captured by subgraph signature function, that is then transferred from one test to the other, essentially from one label set to another label set, in a hope that once we go to our target label set that has very few labels, we will be able to train a classifier despite those few labels in the target label set. That's what GMeta can do by specifying subgraph signature functions that work on top of neural message passing scheme that is the paradigm that kind of powers general GNN architectures.
In a connected question is like, so how does these models that you have generated compare with unsupervised ways of training set expansion, like the ones that have been reported in AAAI 2018, so where the authors use some sort of a Parwalk kind of an architecture and try to expand the training set without any additional information of training levels, and they do it in a completely, based on completely structure guided fashion, exactly like the way that you are trying to find structural signals.
Yeah, so that's a great point. So the way this work relates to the paper that and the research that you are mentioning is really by kind of what is common to them is that they both rely on structural similarity, on kind of recognition that we need to effectively level the structure much more strongly in addition to labels when those labels are limited. So and that is what GMeta does, and I believe that the paper that you're referring to is similar to it in that case, that structurally they rely on graph structural patterns in order to increase the size of the label set. Now, increasing the size of the label set in an unsupervised manner effectively means that we are imputing the labels somehow, and so that might lead to propagation of errors in the underlying model. So an alternative way of addressing the problem of learning in settings where limited labeled data are available are few-shot learning strategies. And so GMeta is a few-shot learning strategy that kind of works in the assumption that we have the very few labels for each of the class that we're interested in, and that's what we want to work with. We don't want to artificially impute labels and then have that artificial label set to train a standard predictor. Though I completely acknowledge that's a kind of orthogonal perspective to this problem.
Thank you very much.
Thank you. So there are two questions. So since it's last, I will just allow. Ajay, I have given you permission to talk.
Thank you, sir.
So you can ask.
Well, my question is, to train, we need a data. It's like wait till virus attack you and then to counter attack, which will of course take more time. So how can we do this in a proper way? My question is regarding COVID-19, and we are having a variant. We have changed ourselves from Delta to Omicron. Thank you.
Yeah,
Did you
so
listen?
it was hard to understand the full question because there was some delay, at least on my end. But if I understood correctly, the question was about the evolving knowledge and the evolving data sets over time, in particular with the COVID repurposing example, where new variants are emerging and we have more information about or potentially even conflicting information about how the virus behaves and so on. So that's not a problem from the machine learning standpoint. That's not a problem for the model, because it is easy to retrain the model from either from scratch on a new data set or continue to train it or fine tune it on a new variant. That is in fact something that we have been very closely looking into and working on, the notion of then not only have a single COVID-19 disease module, but have many different modules that are then specific to different, that are then variant specific. And so that's certainly something that is possible to do. Another point that I'd like to make is that, in particular in the context of the pandemic, there is a number of conflicting information out there, or perhaps it often happens that what preliminary findings identified as promising information later turned out to be not true. And so that means, from the algorithmic standpoint, that we would essentially need to have a model that would be able to delete certain facts or certain predictions from the model over time, as those facts are not true anymore. And so that is also a topic that we are very interested in and are actively working on how one can essentially delete part of the data from GNN models when those in data sets are not, or individual data points are not relevant anymore.
Yeah, thank you. Sai, you want to ask? Sai, you want to ask? I see your... Rohan?
Yes, I want to ask that into the subgraph paper, how have you investigated about the subgraphs through some investigation of the clicks or some any other method? How you determined the subgraphs into that particular work?
So that's a great question, Rohan. So the task there was not to extract or define subgraphs. So the task is given a graph and subgraphs that are given to us. The goal is to learn embeddings for subgraphs. So that is different from subgraph discovery or subgraph detection, or what is known as in classic network science as community detection algorithms that would be concerned with extracting dense sets of nodes from the graph that would be then communities or cliques or that could then be subgraphs. In this particular case, subgraphs are given to us. In concrete example that I was highlighting, each subgraph is a set of phenotypes or a set of nodes, and they are given by the patient representation itself, right? So a disease is described by a set of phenotypes, symptoms that describe the disease, and then that defines the disease subgraph. Because of that, these subgraphs can be very challenging to handle. These subgraphs can be such that each of them has multiple disconnected components, and they're not necessarily densely separated from the rest of the graph, as network communities or clusters would be. So the subgraph neural network work, we were interested in learning with subgraphs rather than extracting or detecting subgraphs in the networks. As for that, there's already quite plenty of existing work, particularly in graph clustering and community detection fields.
Sure. Thank you. Thank you, Marinka, for this great talk.